Welcome again to lecture number 5, and we're going to be talking about statistical spectroscopy. So, just as a quick outline, for today we've looked at both an introduction to metabolomics, NMR-based metabolomics, software, and spectral deconvolution. So for the last part, we'll be looking at statistical spectroscopy. So, as a way of introducing it, and you've seen this slide before, we've identified two approaches to doing NMR-based metabolomics. One, which we call quantitative or targeted, which involves spectral deconvolution. And that means taking your spectrum and identifying specific metabolites and identifying the concentrations, essentially producing a labeled list of data. The other approach involves using a chemometric approach or an untargeted way. There are certain approaches that have been developed over the last number of years called Stocksy and Classy methods. In this case, what you're trying to do is identify specific peaks or specific regions in your spectra that look different when compared to other spectra in your sample set. So rather than labeling or identifying specific peaks, you're going to be coloring your peaks to some extent, with the blue peaks representing those that are relatively insignificantly changed, and the red and orange peaks representing those or peaks that have changed or are changing significantly between groups or cohorts or samples. So these are fundamentally different approaches. In one, with the quantitative method, we're eventually going to be doing statistics with our concentration lists. With the chemometric approach, we're doing statistics with the XY positions of the spectral coordinates. So this is an important difference and something that people need to understand. So in spectral deconvolution, what we're doing is we're taking the mixture and we're identifying individual compounds, we're labeling the peaks, and we're assessing and determining the concentrations. The net result is an annotated list of compounds and their concentrations. In statistical spectroscopy, we're not looking at a single spectrum or even a few spectra. Typically, we're looking at dozens, maybe even hundreds of spectra, and are trying to identify what's different, or what's correlated, or what's not correlated, or what's grouping, based on the features of many spectra when compared. So statistical spectroscopy often begins with stacking your spectra together to look to see how things are aligned and how they look. But it can use or involve many other components to identify interesting features. And again, the role of statistical spectroscopy is not necessarily to identify compounds, but to identify features or coordinates or X and Y positions in the spectra that distinguish several spectra or one spectra from another. So what's central to statistical spectroscopy is you need lots of spectra because you need lots of numbers to perform statistics. So you can't do statistical spectroscopy on one spectrum or 2 or even 10. You generally need to do at least 30 or 40 and generally more. In order to do good statistics with anything, and especially with spectra, you have to be careful about scaling and adjusting the spectra. You have to bin the spectra often or peak pick the spectra, and you have to align the spectra to be sure that you're aligning peaks to peaks or X and Y coordinates to X and Y coordinates. So the central goal in statistical spectroscopy, which is different than deconvolution, is to identify peaks or regions that are statistically different, statistically significant between different groups, within groups, or over time, as measured with this large collection of spectra. If you found statistically significant groups or peaks or regions, then the next goal in statistical spectroscopy is to try and identify the compounds corresponding to those changed peaks. So in some respects, this is almost the reverse of what we do with deconvolution. But the reason why people use statistical spectroscopy is to help simplify the problem. If you know nothing about your system, nothing about the spectra that you've collected, nothing about the compounds that are likely to be in your system, you don't want to spend hours, days, or weeks trying to identify things from scratch. In that case, you would resort to statistical spectroscopy to reduce the thousands of peaks you'll see in an NMR spectrum to a few dozen or even just a few statistically significant peaks. And now the problem of identifying those compounds isn't trying to look at dozens or hundreds, it's now perhaps just a few compounds. So that's the goal of statistical spectroscopy, and it was a noble goal, certainly when it first began, because in most cases we didn't have any idea about what the compounds were in most biofluids or tissue extracts or foods and fluids. So here what we're doing in statistical spectroscopy is we will start with samples, many samples. We'll collect many spectra. We'll not only collect the spectra, we'll scale them, we'll align them, and then we'll perform a statistical evaluation or a data reduction to try and reduce both the number of peaks that we're actually worrying about and identifying those that are significantly different. And here they're colored in orange or yellow. So what might have begun as a spectrum with 1,000 peaks, we're now reducing it to a spectrum of 3 or 4 relevant peaks or regions. And if we focused on those ones, then we can now focus all of our efforts on identifying those significant peaks and what compounds they are. So it defers the compound identification task, and to some extent it can simplify the compound identification task. And in many cases, this approach is widely used in LCMS and even GCMS. In the world of LCMS, we're often dealing with a much more complex mixture, many more thousands of peaks than we might deal with in NMR. But in LCMS, the standard protocols are typically to take lots of spectra, to align them, and then to extract those aligned peaks and identify the ones that are most significantly changed from sample to sample or cohort to cohort. And it's only after we've simplified this collection of thousands and thousands of peaks to perhaps a few dozen that we go to the effort of identifying those peaks and perhaps trying to quantify them if we can. So in the case of LCMS, the use of statistical spectroscopy is well justified because the number of unknowns is so large. And in the early days of NMR, the use of statistical spectroscopy was also well justified because in those days we barely knew what was in a typical blood or urine or serum sample. Now today we do know that, and in that regard the need for statistical spectroscopy, at least for NMR, is not as great. But in LCMS, where there's still up to 95% unknowns, the need for statistical spectroscopy is more strongly justified. Now, I have a strong bias. I believe that spectral deconvolution is more appropriate. In fact, even the people who developed spectral or statistical spectroscopy have now largely shifted towards spectral deconvolution and targeted identification. But for this lecture, we're going to talk about it because it's historically important and because there's still a number of people that use this. And there are certain situations where statistical spectroscopy can be helpful. So there's several steps for statistical spectroscopy. The first part involves spectral collection, which is not unlike any other beginning metabolomics experiment. As with almost any metabolomics experiment, you want to collect a large number of samples that might represent 2 different cohorts or 3 different cohorts, or they may be over different time periods. In statistical spectroscopy, more samples is better because you're trying to make use of the power of statistics to look for possible correlations or connections. What you also have to do with statistical spectroscopy is be very, very careful about how you collect and process the NMR spectra, often much more so than you do with deconvolution methods. You want to make sure that samples are prepared as uniformly as possible, as identically as possible. Also, to prevent any bias, you want to randomize the sample order. In general, you want to use the SOPs or protocols that we described in Lecture 2 that talked about some of the specific acquisition parameters and techniques. But key, again, in statistical spectroscopy is to make sure that the samples are as identical or near identical as possible. And that's generally good practice regardless of whether it's statistical spectroscopy or regular deconvolution. So if this is a sample of plants, we would try and collect 50 or 500 or 5,000 samples, process them identically, and collect 5,000 samples, collect 5,000 spectra in a manner that is as similar and as identical as possible. After you've collected the spectra, you'll do standard processing, pretty much as we've described again in Lecture 2. You'd be doing zero filling, Fourier transform, phase correction, baseline correction, solvent filtering. The key, of course, is to apply these processes identically for all of your spectra. So there's no variation between them, and that you can be sure that there's no introduced bias because of changes or modifications to your spectral processing conditions. So at this stage, there's a lot of similarity between statistical spectroscopy and classical deconvolution methods. The second step is where things start to get a little different. And typically, because you're trying to make things as identical as possible, you often have to do a careful job of scaling or normalization. So this might be an example where you've collected a bunch of spectra, and you can see that for the most part, the peaks are in the same position, but the intensities of these peaks vary quite a bit. Now, some of that variation may be real, but it might be also a case that it's due to things like dilution problems or sample weighing or sample preparation. And in that regard, you want to be able to adjust or rescale these diluted spectrum or overly concentrated spectra so that things are more in line with each other. And this is, again, why constant sample preparation and control of homogeneity and constancy is critical for statistical spectroscopy, because if they aren't, you will start to detect artifacts, and the artifacts will be amplified through the statistical processes. So if you're scaling things, it certainly will ensure your intensities are relatively similar from sample to sample. So you're not comparing something that's been diluted by a factor of 10 with something that hasn't been diluted at all. So in other words, you're trying to compare apples to apples and oranges and oranges. You're trying to correct for sample variation that can happen randomly or biologically, where things can be diluted or concentrated. A good example is urine, where depending on how much an individual has been drinking, they can actually dilute their urine quite substantially. It's also an issue for things like fecal water or food extracts, where things can be more hydrated or less hydrated, and simply weighing the wet weight is not a good indication of how much dry matter is being added. It can also be a problem for cell extracts and tissue extracts. And these issues are not only problematic for statistical spectroscopy, they also need to be addressed when you're doing spectral deconvolution as well. So one of the ways, or there are actually several ways of scaling spectra, or ensuring that things are roughly comparable. One way of doing that is to add a spiked standard, a special compound, probably not, or generally not a metabolite, but something synthetic. It's added at the very beginning of your sample preparation protocol. And that way you can compare sample to sample to sample and see that that exact compound and the exact concentration is still the same. And that way you can ensure that you're scaling appropriately. You can also scale to an internal standard. This is commonly done for urine. It's been done for decades. And the compound used there is creatinine. And that's why most urine concentrations are quoted with reference to creatinine. Another technique that's commonly used both in statistical spectroscopy, both in NMR and in mass spec, is to pool your samples, to take a small aliquot from each of the 50 or 500 or 5,000 samples and make a super sample that has a bit of every component of your sample set. This creates an average, if you want, and allows you to essentially compare against that average set. You can also scale against the total integrated area, at least in NMR, which represents the organic content of the sample. And this corrects for, again, dilutions or concentration events. Or what's even simpler than that is you can scale to density or refractive index. And there are simple and inexpensive pieces of equipment that can measure refractive index or density. And again, this helps you to ensure that all of your concentrations have been properly adjusted so that the set of spectra you're working with is roughly comparable. Now, this scaling is done not only for statistical spectroscopy, but it can and often is done for deconvolution as well. Now, there's a difference between what I call sample scaling and data set scaling. So sample scaling is what we've just talked about, is essentially trying to correct for known or expected technical or biological variations. So these can cause or lead to an inadvertent dilution or an inadvertent concentration. So we're just trying to make sure that everything is roughly comparable, and that a weighing mistake or a processing mistake or a sample that's been processed differently or handled differently doesn't throw things off. Another thing that you do is only when you have your complete set of spectra, and this is called data set scaling, or it's also called normalization in other cases. And this is when all your data have been measured, and you're now trying to adjust for general differences in the overall concentrations or intensities of different peaks or features or bins. Technically, what you're doing here is you're adjusting for fold differences in the concentration of metabolites. Some metabolites will be highly concentrated, and because they're so dominant in their intensities, will outweigh statistically all the events that are happening with the less abundant or weakly concentrated metabolites. So this is something that's critical. It's essentially you don't want the loudest voices to win over the weakest voices. In some cases, the whispers coming from weak metabolites may be just as important. Just as the weak signals from low abundance proteins and genes are often very important. So what you try with data set scaling when you're working with all of your spectra is to convert the differences in relative intensity or concentration to scaling factors so that you inflate the small values so that they are not lost, so that they're roughly comparable to the big values. Now, if you were just randomly or characteristically all small values that are as big as the big values, you will cause some problems. And this is because small values, weakly low intensity peaks, or low intensity signals typically have a bit more noise. And so their accuracy isn't particularly good. So there's an error that will be introduced. So what you want to do is not only scale by a general number, but you also want to divide by a number that statistically characterizes the variation in those peaks or their concentrations. So in terms of data set scaling, there are at least 4 or 5 different options. Auto-scaling, Pareto-scaling, fast-scaling, scaling by average, or centering. So the correction factor that we use in auto-scaling is to use a standard deviation. So that way we'll introduce or ensure that all of the peaks or features, or if we have metabolites, all have a standard deviation of 1. With Pareto scaling, we'll scale by the square root of the standard deviation of the scaling factor. In vast scaling, we'll use the coefficient of variation, which is often called CV, as a scaling factor. We can also scale by average, or the mean of these samples. And the other route is by centering, where we can convert all of the values so that they're centered about 0, so that things are positive and things are negative. All of these are possible, but there's a general preference to use either auto-scaling or Pareto scaling. So these are critical. These are techniques that are also used in deconvolution methods or quantitative metabolomics, but these are particularly important for statistical spectroscopy. Another component or another step that's really unique to statistical spectroscopy is something called spectral alignment. And this arises when spectra are collected, and either due to temperature variations, pH variations, salt concentration variations, human errors, that the positions of the peaks are varying from spectrum to spectrum to spectrum. Now, this is a particular problem with LCMS and GCMS-based metabolomics. It's less of a problem for NMR-based metabolomics, but it still is a problem. So this is an example of what one might see, where if you're collecting many, many spectra and you have everything nicely scaled and you have everything beautifully oriented, everything's been collected very precisely, and what you see is a whole bunch of peaks that are moving back and forth. These doublets that we're looking at, and we can see that they're shifting. And clearly it's the same compound, but for whatever reason they're moving. And so this will mean that if you're to do statistical comparisons against these peaks or these bins or these XY values, you'll come up with very misleading answers. So what you have to do is then force those peaks into alignment. This is a spectral alignment process. Now, this really does occur, and this is a real example of a urine sample where we're looking at citrate. Citrate's characterized by two doublets, one at about 2.35 ppm, one about 2.7 ppm. We're looking at 4 samples, genetically identical mice, their urine, and at essentially identical pH, 7.4. But if you look closely, you can see that these doublets actually are shifting relative to each other. If you compare the purple to the blue to the green, you'll see that they're shifting in some cases by about 0.02, 0.03 ppm. We can also see that the line widths are varying as well. The one at the bottom has a wider line width than the ones at the top. So these are all effects that can arise from the matrix and can lead to spurious statistical results. So again, you want to make sure that all the peaks are aligned. As I'd mentioned before, peak positions can be affected by pH, salt, temperature, by shimming, diffusion, chemical exchange, human or instrument errors, general matrix effects, also chemical shift referencing errors. All of these contribute. And so for doing proper statistical spectroscopy, you want to make sure that those are dealt with through spectral alignment. Just like the scaling that I'd mentioned before, spectral alignment is also essential because it ensures you're comparing the same peaks from sample to sample. Again, it's apples to apples and oranges to oranges. So if you're comparing peaks to noise, then you think you probably found something significant. But if you're comparing peaks to peaks, then in fact maybe that's not so significant. So as I say, this corrects for spectral misalignment, and it's widely used in NMR-based metabolomics, LCMS-based metabolomics, and even GCMS-based metabolomics. Now to do spectral alignment, there are several routes that you can do. In the earlier days, when computers weren't so fast, people would align by bins. They would go through the spectra and draw squares or rectangles around all of the regions. This effectively reduces the resolution, but it makes the calculation of alignment very fast and very simple. Another approach is to align by peaks. And this is more intuitive because people think in peaks. But typically to identify peaks, you have to do some peak picking, and humans are generally better than computers. What's more, peak alignment generally requires more computing. Another technique is to use something what are called correlated spectral intervals. And this allows you to work at generally higher resolution than binning. It can be automated. It's also very fast. So we'll just talk about binning because it's a little more intuitive. And as I said, it was the very first technique that people used. So here's an NMR spectrum, and essentially we've drawn bins of roughly about 0.1 ppm width around different regions throughout the spectrum. The bins cover essentially peaks and in some cases noise. Some bins have essentially no spectral information or just noise. Others have a few peaks. What we'll do is we'll measure the area under the peak and define the area under the peaks or above baseline as the volume or area, or give a number to that particular bin. So in this way we can convert 1, 2, or 3 peaks, or tiny peaks and big peaks, into one value, which represents an area. So this is an example where we've essentially binned a spectrum on above, which is shown below. And basically you can see that the bins generally correspond to peaks. Some areas you can see where, especially around 3.5 to 4, the bins seem to be quite a bit bigger than the peaks. In other cases, peaks seem to be quite a bit smaller than the bins. And this is just simply a consequence of the resolution loss. But in essence, what's happened is we converted a spectrum that had 60 or 70,000 points to a spectrum that maybe has 100 bins. So that makes it a whole lot simpler. And then we can do something that's relatively easy. We're looking for the bins that should be aligned, where they're different. And if you compare these blocks or bins or rectangles, you can say for the most part they match, but what I've marked in red are bins that don't match. And so all we have to do is shift the bins left or right so that they do match. So the binning alignment algorithm, as I say, is relatively simple. You compare each bin spectrum perhaps to a standard, one that you've chosen, or maybe an average one. And then you match each bin based on its area and position of the query spectrum to the standard. You'll adjust the positions of those bins left or right by 1 or by 2 until you finally get a good match where the intensities and positions seem to be good. Obviously, if you made really large bins, generally you won't have to do a whole lot of alignment. If you have really narrow bins, then sometimes you have to do very fine alignments. Now, binning automatically leads to a loss of resolution, and some of the fine structure and couplings are lost. So if you can measure at the level of peaks, then you have more of that resolution, and obviously contain more fine structure that's chemically important. So in some respects, peak picking is similar to binning. It's just to a much higher resolution. So in this case, we can take an example where we have a spectrum, say, with great-looking Lorentzian peaks and essentially no noise, and it's very easy to convert those peaks into bins and to match them with specific intensities. That's easy, but this is an example of a hard peak picking exercise. There's a lot of noise, and in fact, most of us would probably argue that there are no peaks in this particular spectrum. So peak picking is not easy. Humans can do a better job of computers. But if you can do some decent peak picking or are willing to do that, you've now converted your spectrum and now you can align peaks very much the same way that you would have aligned your bins. And the alignment algorithm is very similar. You identify your position of the peaks and their height, their width, and their position. And once you've got that, then you can do an alignment where you're matching the standard spectrum, the peak picks, to the queried one, and align things up to a certain level of parts per million in terms of maximizing the match. And you repeat for all of the spectra and all of your samples until all of the alignment is completed. This approach, whether it's through bins or through peaks, actually is very much similar to the way people do sequence alignment. And this is called dynamic programming, but it's also, for peak alignment, it can be called dynamic time or dynamic frequency warping. So you can use a long-standing, well-known algorithm called dynamic programming that's been around since the 1960s to get that matching, and it can be reasonably fast and very consistent. So this is what's been used for a while, but more recently, a much more elegant approach called ICO shift has been developed. And this is an example where you've taken a spectrum where there's clearly variations in pH. There's many, many samples, and that's the top one. Using ICO shift, it's possible to get all of those spectra that are all over the place, all nicely aligned. It's very fast. It was developed by a group in Denmark, and it's one of the tools that's used in MVAPAC, developed by Rob Powers. So in this case, ICO shift is essentially sort of an iterative method for looking at spectral intervals and looking at correlated shifting of those intervals. To make it fast, it uses a fast Fourier transform that allows you to do this alignment simultaneously. So it's actually much faster than other methods, and it makes use of full resolution data so you don't have to do the binning, so you don't lose your resolution. So the program's, as I say, available and can be freely used. So in that way, now ICO shift has largely become the standard for a spectral alignment, particularly for NMR. So if you've done your alignments and you've done your scaling, the data scaling, the spectral scaling, you've collected things very carefully, now you can start doing the statistics. And the statistics here are to try and look at how the peaks or the features or the bins are differing from sample to sample to sample. In this case, what we're going to look at, or we're trying to use, is multivariate statistics to see where those peaks are most significantly varying. And what's shown on one side is called a principal component analysis, and what's shown on the other side, in part B, is the information plotted out in a regenerated NMR spectrum, which is showing positively changed features pointing up, and the negatively changed or negatively correlated features pointing down. So as I say, once all those spectra have been collected, aligned, sorted out, we use multivariate statistics to analyze the data points of the spectra. We're not analyzing the metabolites because we haven't identified. We're not analyzing their concentrations because we haven't deconvoluted them. We're just treating the spectra, the XY values as the spectra, as the inputs to our multivariate statistical tools. In effect, we're looking for outliers of data or differentiating peaks or bins or collections of peaks. What you can do is actually then recast or regenerate a pseudo-spectrum that can be plotted out, which shows essentially the correlation coefficients or regression coefficients plotted against parts per million. We'll talk about PLSTA and PCA in lectures 11 and 12. So with the statistics, with these statistical tools, the multivariate statistical tools, we can look at a collection of 100, 1,000, 10,000 spectra, and we can see, in one case, the outliers. Of course, this is obvious to your eyes, but if we were looking at many, this is something that statistics could pull out. Another thing we could do with the statistics is perform principal component analysis, where we're trying to distinguish between, in this case, two groups, blue spectra from the red spectra. In this case, the blue spectra might correspond to healthy and the red might correspond to diseased. And if we look at the things marked in these boxes, we can see that in some cases the peaks are varying. In the blue spectra, the doublets generally seem to be slightly lower intensity relative to the red spectra, which generally seem to be of higher intensity. On the other hand, if we look at the singlet marked in the green box, we can see that the intensities for the blue samples are higher, whereas the intensities for the red samples are lower. Again, it's obvious to our eyes, but principal components analysis could look at hundreds, thousands of data points and pick out other trends that might be more subtle or not easily detected by our eyes. So in this regard, we can do multivariate statistics. We can identify those differences, and then we can highlight them in these graphs that show the variations and the degree of correlation or differentiation. So we've regenerated the spectrum, regenerated the plots, and in many cases we'll color those plots to show where those variations are greatest. There's another thing you can do beyond just simply looking at that principal component analysis. We can do analysis of statistical techniques to look for covariance within the whole spectral data set. So not differentiating between group A and group B or red and blue. In some cases, we can use this approach to identify highly correlated, very strongly correlated peaks that change from sample to sample with exactly the same magnitude and exactly in the same direction. Or we can look for weakly correlated peaks, which may not necessarily change in exactly the same magnitude or direction. So in that first scenario, here's an example. We're looking at maybe 100 different spectra, and we might be looking at just one particular case or over different times. What's highlighted here is that you can see that the doublet on the left and the singlet on the right for sample 1 are very high. For sample 3, the doublet is very small and the singlet is very small. And at sample N, the doublet is high again and the singlet is high. So what you're basically seeing is that there's a very strong covariance with these three peaks. Therefore, they must belong to the same compound. So that's an important thing that you can get by looking at statistics. Now, of course, we could do this using COSY or TOXY and see that, in fact, there's coupling between these two. But if we are only doing 1D, this effectively is giving us a statistical TOXY. Another approach is to look at weakly varying things, where, again, sample 1, the peaks are high, the doublet and the triplet are high. In sample 3, the doublet and triplet are low, but in fact the doublet is very low compared to, say, sample 2. And then in sample N, the doublet is relatively higher than the triplet, but they're still diminished over what they were in sample 1. So in this case, the peaks are sort of covariant, but they're clearly not the same compound. They may be in the same pathway. So these are the two types of correlations, the two types of scenarios you can get in metabolomics, or at least with these statistical spectroscopies. And so over the last decade or so, people have formalized these relationships. The first one to appear was something called stoxy, or statistical toxy, which essentially is a way of pulling out those covariant peaks that allow you to identify specific compounds without actually having to do a complex two-dimensional experiment. Another method called classy was developed in 2009, storm developed in 2012, and stoxy developed in 2014. I'm going to talk about stoxy and classy a bit more, and just essentially as examples of statistical spectroscopic techniques. So stoxy was the first one, and it was really used to help identify individual compounds that happen to be covariant from complex 1D spectra. It's a statistical version of toxy. It allows you to identify coupling patterns if you want, because you're looking at multiplets that seem to covary. You can plot out a stoxy just like you would in a 2D toxy, or you can convert it to a 1D version, coloring it from red to orange to yellow to light blue to dark blue in a heat map. So in stoxy, you're basically using the linear relationship between intensity that belong to the same molecule in an NMR spectrum. So by looking at how those intensities covary with each other, whether they're peaks or bins or spectral points, allows you to create a correlation matrix. And that way you can present a pseudo or fake NMR spectrum that shows those correlations. So in some cases, if there's very strong correlations, those are the same molecule. If the correlations aren't so strong, those are probably related metabolites that might be in the same pathway that are being up or down regulated. So you can then plot the correlation of those resonances by a color scale, where there's low correlations might be dark blue, and high correlations, which will have a value of 1 or close to 1, will be red or orange. So this is just illustrating what a two-dimensional stoxy picture looks like. It looks a lot like a COSY, or double quantum filtered COSY, or even more detailed toxy. These are just simply illustrating the correlations and chemical shifts between different peaks. So you can see there's a diagonal, and then there are off-diagonal elements. This is actually hard to interpret. So what most people do today is produce the one-dimensional version, because in fact that's typically what you're measuring. You're getting a 1D spectrum. And by plotting those that are highly correlated, that is red or orange, those peaks that are not well correlated as blue, you can identify in this particular case 4 sets of peaks that are highly correlated. One set of peaks, a triplet at about 2.5, another which looks also like a triplet around 2.9, and then you can identify 2 or 3 doublets, one at 7.4 and one at 7.2. These all belong to the same compound. And by looking at the spectra carefully, in this case, they actually isolated the compound. They were able to identify it as a sort of a phenolic acid in the compounds depicted there. So potentially you could have identified this using toxy or through standard isolation methods. But this essentially was identified as being A, an important metabolite because it varied a lot, and B, using the chemical shifts and their covariance, you're able to identify that this belonged to the same molecule and was sufficient to start identifying a probable likelihood. A, it's an aromatic molecule, slightly asymmetric, and B, that there's probably some aliphatic portion, which is in this compound. Classy is a later version of NMR statistical spectroscopy. 2 different variants were sort of developed by 2 different groups. One was to try and again identify the same molecule via the correlation of spectroscopic variables. The other one was to look at clustered peaks using the ratio of covariance and a correlation of the variables. So this is described after stoxy. It can be done both in 1D and 2D, and this is an example of a classy NMR statistical spectrum. So statistical spectroscopy is possible, it's feasible, it's supported by a number of tools. Perhaps the most easily available one is called MVAPAC, which is a downloadable software package offered at the University of Nebraska in Lincoln. There's another software package, it's also downloadable, called Atomics. You can also get tools that support aspects of stacking and alignment and some aspects of statistical spectroscopy in MNOVA, AMIX, and Cannomics. Those are commercial. And MetaboAnalyst will also support some aspects of statistical spectroscopy. Now, I'm talking about statistical spectroscopy partly for historic reasons, because it is not an area where there's a lot of advancement, and it's one area which I think is largely getting replaced by spectral deconvolution. However, it does have some advantages. It's ideal for handling very, very large sample sizes or sets, so tens of thousands of samples. That's generally too hard for an individual to deconvolute using software like Cannomics or Amix or even Basel. So in that case, you might want to use statistical spectroscopy. In that regard, if it's done well and programmed well, you can automate components of it. The fact that NMR is very reproducible, instruments are highly reproducible, things relatively very little, is a real strength for this. And in many cases, it allows you to get information sometimes that's very complex. So if you're dealing with a sample that you've never seen before, from an organism you've never worked with before, this might be a good first start, at least to identify what's important or what's not important. It's a relatively unbiased approach at looking at NMR spectra. However, because it's so dependent on having things, especially peaks aligned or bins aligned, of having everything properly scaled, of having carefully controlled environments, carefully controlled SOPs or standard operating protocols, it's often prone to artifacts and false leads. It also delays the inevitable, in that you ultimately have to get compound identification, compound quantification. If you don't do that, then often you can't publish, you can't do anything that's biologically useful, can't make any biological interpretation. So in that regard, deferring, delaying, or completely avoiding compound identification and quantification makes a lot of the work that you do in statistical spectroscopy somewhat wasted. Many cases, people use statistical spectroscopy because they don't want to try and buy or purchase or assess the authentic standards to verify what they think is there. In some respects, statistical spectroscopy, although pretty, can be quite hard to interpret. They're not clean spectra. They're statistical spectra. What you get from statistical spectroscopy may identify important peaks, but those peaks can't be used in clinical applications or standardized testing or reference work. And as I said, over the last 5 years, statistical spectroscopy is largely being replaced by spectral deconvolution methods, which are becoming both faster, more robust, given the fact that most metabolites in most kinds of NMR spectra can be relatively easily identified, and most are well known. Now, I have a pet peeve, I guess, about statistical spectroscopy, and I think it's important to understand. One is that statistical spectroscopy is not inherently bad, but it was developed at a time when very little was known about the composition of the metabolome, when we didn't know what the composition of blood or urine or saliva was, when we didn't know about the composition of wine or juice or honey. Now those things are known. And in that regard, there's no need to try and do statistics on something that you already have a good idea of what the composition is. And in that regard, spectral deconvolution is ideal for these. Statistical spectroscopy also emphasizes statistics and chemometric methods over good analytical chemistry practices. Now, this can be something that you'd use when you are sort of up against a wall or stuck in a corner, or where you're dealing with something where no one knows absolutely anything about the mixture or the sample. But a good analytical chemist will want to know what they're measuring. A good analytical chemist will want to identify and quantify as accurately as possible. And that's what spectral deconvolution does, not statistical spectroscopy. It's very hard to publish results if you don't identify and don't quantify what you're seeing. And so again, in the end, whatever you do, you're still going to have to identify and quantify. Most of the field of statistical spectroscopy grew out of work that Jeremy Nicholson pioneered, especially in the area of metabonomics and toxicology. And many of the people who worked in his lab have largely adopted it. But even the original lab that started it is now largely moved to targeted metabolomics using spectral deconvolution or targeted MS, where compounds are identified and quantified first before the statistical tests are applied. So with that, that's my story on statistical spectroscopy.